Okay. Hello. Good morning. Well, we are going to start the app of the defense. Well, before to start, we would like to do some comments. The defense is divided in two parts. In the first part, the candidate will present the work carried out during her PhD, and the estimated time is about 45 minutes, more or less. In the second part, the candidate will have a debate with the member of this committee, lasting approximately 60 minutes. We can see. And the public will then leave the room, and the member of the commission will discuss the presentation and defense in order to agree on the qualifications. And afterward, the candidate and the public will return to the room, and the commission will inform of his qualification. In this act, we will give you only the qualification of this part. The other part is about the extraordinary hour will be obtained after a few days from the university, because we will need to voting in the case that you have the high qualification. I would like to, before to start, I would like to introduce the member of this commission. On the left, Dr. Michel Groff of the Catalonian Water Research Institute. On the right, Professor Fiona Regan from the Dublin City University, who is speaking, Julian Blasco from the Spanish National Research Council. And now we are going to start without delay.
So, good morning, everyone. It's very special to be back in Girona and be accompanied today by friends, colleagues, and family. So thank you all for being here today, and I hope you enjoy the presentation. Thank you also to the committee for accepting the invitation. So, this thesis was conducted, as you know, at the Catalan Institute for Water Research under the doctoral program in Water Science and Technology from the University of Girona. This is the title of my thesis and also of my presentation today: Biomonitoring of Pharmaceuticals in the Mediterranean Aquatic Environment, an Interactive Effect with Microplastics, Insights from Field and Lab Studies. In this presentation, firstly, I would like to introduce you to the background of the study. Then I will present the main hypothesis of the work. Then I will go through some of the main outcomes of the study, which are divided into 3 different chapters. And then I will proceed to read the main conclusions of our work. So, Mediterranean aquatic ecosystems are threatened by a series of natural and anthropogenic factors, such as water scarcity, the overexploitation of resources, changes in land use and direct deterioration of habitats, aquatic pollution, and the typically limited dilution capacity of our streams, and the influence of certain extreme events, such as flash floods, which can increase the release of pollutants into the receiving environment. These extreme events can increase in frequency and magnitude under climate change scenarios. And altogether, this can lead to a loss of water quantity and quality, which is essential for the preservation of ecosystems and also for water safety for human use. These images show the degradation of one of our study areas, the Mar Menor Lagoon, which has changed a lot in just a few decades. And I will introduce you to this study area a bit later. This table aims to show some of the main contaminant groups that can be found in our rivers, streams, coastal areas, or wetlands. Of course, these are not unique to the Mediterranean area, and their importance will depend on the predominant sources in each place. But just to show you that some of the selected contaminants for this thesis are tightly connected with urban discharges of wastewater, such as pharmaceuticals and personal care products, also microplastics, but other sources might have a contribution as well, such as agricultural inputs. focusing on pharmaceuticals, these substances are considered to be emerging in the environment because typically they have been subjected to a limited regulatory framework, and also they are considered pseudo-persistent, not because they are very stable per se as substances, but because they are continuously released into the environment. This has led to a widespread presence of these compounds in aquatic ecosystems. Once they are released, they are subjected to different environmental processes, such as dilution, phototransformation reactions, sorption on particulate matter, or accumulation in organisms. Several therapeutic groups of pharmaceuticals can be found in these environments, such as anti-inflammatories, antibiotics, antihypertensives, psychiatric drugs. And these substances can cause effect in non-target organisms. The kind of effects that they cause can follow the mode of action that these pharmaceuticals have in humans, but sometimes they might lead to certain unexpected effects as well. They have been subjected to a limited regulation in the environment. In general, there's a lack of environmental quality standards. However, in the last decades, some of these compounds have been included in watch lists at European level to facilitate their monitoring in European water basins. Some recent regulation on water reuse, for example, also mentions pharmaceuticals as part of different emerging compounds that should be considered when authorizing the reuse of water for different purposes. And very recently, just a few weeks ago, a new wastewater directive came out, and for the first time includes pharmaceuticals among the different types of contaminants that should be efficiently removed from wastewaters in order to protect the environment. There are different approaches to study the presence of pharmaceuticals in the environment, different aspects that should be taken into account when you study the occurrence of these substances. For example, when you go to the field, you need to select a suitable study area, depending on the objectives of your study. You need to collect different types of samples, and you need to select the right sampling time, also depending on the purpose of your study, different seasons, or taking samples just after a flood that you suspect to be relevant in terms of pollutant discharge, for example. Typically, once you collect your samples in the field, you take them to the lab, and then you will apply a methodology which is generally adapted to your sample type, but you don't treat the same way water samples than fish samples. But one thing that these methodologies have in common is that they need to pre-concentrate the contaminants, because they are present at such low levels in the environment that you need pre-concentration techniques to obtain the final extracts, which are the vials that you take to your instrument and analyze. So when you have a final extract, you can apply different types of analytical approaches, generally based on mass spectrometry, because you need a very sensitive technique to detect these compounds, but different approaches are available. In this table, I try to summarize the different approaches which are available: target analysis, suspect screening, or non-target screening. They differ in the type of instruments that are generally used, the requirements for reference standards, the type of results that they are providing. In general, the more you go to the right in this table, so towards non-target screening, the higher the chemical coverage, but also the higher complexity in the treatment of data and in the identification process. So depending on the purpose of your study, it might be more convenient to choose one approach or the other, or even combine them. So switching to plastic pollution, as you know, plastic production is huge nowadays, and it's expected to continue increasing. By definition, microplastics are simply particles of plastic smaller than 5 millimeters, but within this very heterogeneous group of contaminants, different types of polymers, sizes, or shapes can coexist. Once these contaminants are released into the environment, they are subjected to a complex environmental fate, which I'm trying to illustrate here. According to their origin, microplastics can be divided into primary microplastics or secondary microplastics. Primary microplastics are those that are intentionally produced small for a given purpose. For example, microbeads in cosmetic products, or plastic pellets used in manufacturing processes. while secondary microplastics are those that come from the fragmentation of larger items of plastics and fragment in the environment due to the action of UV light, mechanical action, and microbial activity. These microplastics can be ingested by certain organisms and be transferred through the food chain, causing some effects as well. These microplastics can be colonized by microbial communities through biofouling processes, which can change the fate of these contaminants in the environment because they tend to sediment to the benthos, and under certain conditions they can be resuspended as well. Also, the surface of plastics can absorb certain chemical contaminants that are also present in the water column. So as you can see, the fate of microplastics is very complex, and throughout this thesis, we are touching some of these processes. In regards to the interaction with organisms, plastics can lead to different types of effects. The macro effects would be related to large plastics, such as entanglements or digestive blockages, while micro effects are related to the ingestion of small particles of plastic. Typically, after ingestion, certain inflammatory responses can occur, which can lead to the generation of oxidative stress, and this can lead to implications for the whole metabolism of organisms. Due to the suspected ecotoxicity of microplastics, certain regulatory measures have been applied lately, such as the ban of single-use plastics or the ban of primary microplastics in cosmetic products. Generally, these measures try to avoid to fight microplastics from the source, because once they are released into the environment, it's very difficult to get rid of them. So regarding microplastic effects, some of the effects can be related to the microplastics as particles. These are the direct effects of particles. Also, effects can be related to the leaching of certain additives, because plastics are not only the structure, the polymer, but a combination of different substances are added during manufacturing, and this can leach from the plastic and affect organisms as well. And also, you have effects related to the sorption of other chemical compounds, not only chemicals, but also biological agents that adsorb to the surface of plastics and can get in organisms thanks to the plastics, let's say. So on the one hand, the effect of biofouling processes, and on the other hand, the adsorption of chemical agents. The adsorption of chemical agents can lead to a phenomenon known as Trojan horse effect, which is basically consists in the adsorption of these compounds on the surface of microplastics and their transport into organisms through the plastics. Inside the organisms, these compounds can be released. The magnitude of this effect depends on different factors, such as microplastic and chemical specific properties, the characteristics of exposed organisms, or the environmental conditions. The relevance of this effect in general has been questioned, and most of the existing studies are focused on hydrophobic compounds because it's suspected that they can have a higher tendency to adsorb on plastics, but little information is available on this effect in more polar compounds such as pharmaceuticals. In this review, I tried to summarize the existing evidence on the effect of microplastics in the transport of pharmaceuticals and personal care products, or PPCPs. The y-axis shows the ratio between bioaccumulation with microplastics divided by bioaccumulation without microplastics. So if this value is above 1, it means that microplastics are actually facilitating the transport of these contaminants into organisms, while if values are close to 1, this would be a negligible effect, and lower values indicate a cleaning effect of microplastics, which can also occur. As you can see, there's a limited amount of studies, taking into account that there are several pharmaceuticals available in the environment. Most of the studies report vectoring effects because they are above 1, but there is controversy on the relevance of this effect for these compounds. So, to assess the effects of these compounds in the environment, there are different types of approaches. On the one hand, classical approaches typically rely on the measurement of few apical endpoints in organisms, such as mortality, reproduction, or behavior. These toxicity parameters are typically used to derive predicted no-effect concentrations, or PNECs. These PNECs are the basis to estimate environmental quality standards, which are the basis of regulation to protect the environment from the effect of these compounds. And also PNECs are the basis to estimate risk quotients, which give you information on how risky a compound can be in the environment according to the measured or predicted concentrations of this compound in the environment and its toxicity. So values higher than 1 typically associate with a high risk of this compound in the environment, whereas lower values indicate either a low risk or no risk at all. On the other hand, advanced ecotoxicological approaches rely on the measurement of multiple molecular level responses, and this kind of information is typically provided by omic tools, which have emerged in the last years with great importance. In this study, we focus on one of these tools, which is known as metabolomics, and consists in the measurement of the complete set of metabolites in a given organism or community. This approach is typically conducted using high-resolution mass spectrometry, although all the other instruments can be suitable as well, through targeted or untargeted methods. These allow the discovery of new biomarkers, for example. They provide information on early responses that occur in organisms. Therefore, this type of information is mechanistic. It provides information on how this chemical interacts with an organism and can cause effects that are more easy to see, such as mortality or reproduction. So at the end, these early molecular responses are connected to apical endpoints, which are more relevant to regulation, for example. So in classical approaches, typically very well-defined model organisms are used, typically corresponding to different trophic levels, such as microalgae, daphnia, or zebrafish, which are typically used in standard ecotoxicity testing, while advanced approaches can include other models which can be suitable depending on the purpose of your study. For example, in our experiments, we selected river biofilms. There are also marine biofilms available, but we focused on river biofilms as models in freshwater systems. They can be good passive samplers of contaminants because they are sinks of certain compounds in the environment, while on the other hand, marine mussels have been more historically used in the monitoring of compounds. They are active filter feeders, and through this mode of feeding, they can concentrate a lot of contaminants present in the water column. Additionally, they share a set of common characteristics which makes them interesting as models, such as the fact that they are widespread in the environment and they are quite easy to collect. They don't move, they are associated with substrates. They can be reservoirs of antibiotic resistance, both of them, and in general they are good indicators of ecosystem health. So we are moving to the main objectives. I will start with the initial hypothesis. First hypothesis was that the use of multiple environmental matrices and advanced screening methods could improve the assessment of pharmaceuticals in the environment. And second hypothesis was that microplastics interact with PPCPs and increase their bioaccumulation and ecotoxicological effects. To address this hypothesis, we set the 3 different specific objectives. First one was to investigate the sources, fate, and temporal trends of pharmaceuticals in 2 vulnerable aquatic ecosystems. Secondly, to investigate the performance of classical and advanced screening methods for pharmaceutical monitoring in the environment, also addressed through field study. And finally, to investigate single and combined effects of microplastics and pharmaceuticals and personal care products on aquatic organisms. And this was assessed through 2 different lab studies, which I will introduce later. This figure tries to summarize the different approaches that I applied in this thesis. In the upper part of this figure, you can see the 2 field approaches, one focused in the Ebro River Delta, and the other focused on the Mar Menor Lagoon. In the bottom part, you can see the lab approaches through 2 different experiments. In the left side, you can see approaches related to the freshwater environment, and in the right side, you can see approaches more related to the marine environment. So I will be using this figure to try to guide you through the presentation of the results. So starting with chapter 1, focused on the sources and fate of pharmaceuticals in aquatic environment. So within this figure, we will be focusing on the upper part because we will be touching the outcomes of field studies. So the fate of pharmaceuticals, the temporal trends at different levels, or the impact of a specific flash flood that very seriously affected the Mar Menor in 2019. So the two field areas that we selected for this study are located in the Western Mediterranean, one of them in the northeast of Spain, the Ebro River Delta, and the second in southeast Spain, the Mar Menor Lagoon. I will introduce them separately now. The Mar Menor Lagoon is considered the largest hypersaline lagoon in the Western Mediterranean. It is semi-confined from the open Mediterranean Sea, only connected to it through few channels. You can see in these images that I already presented before, the speed of sand that separates the Mar Menor in the right from the Mediterranean Sea in the left. This speed of sand is known as La Manga, and it has been seriously disturbed in the last decades by heavy urban development. Other factors have affected the lagoon as well, which I'm trying to summarize in this timeline. So from the 60s, when we had the virgin lagoon, in the 70s, different changes in land use, most importantly, the change from traditional rain-fed agriculture to intensive agricultural practices has had different implications, such as the intensive use of fertilizers, for example, which lead to eutrophication processes in the lagoon. In the 80s, there was a boom of heavy urban development and tourism in general, so the type of changes that you can see in the above picture in the area, also with implications in terms of wastewater releases into the lagoon, for example. Also in that time, the first wastewater treatment plants were constructed, and then in the beginning of the century, these treatment plants started to be upgraded. In 2010, the first studies on pharmaceutical pollution in the area took place, and between those studies and our studies, some things have changed in the area. Most importantly, the management of waste waters, because since 2015, the treated wastewaters generated in the area are fully reused for agricultural purposes. So we suspected that this could have an effect on the discharge of wastewater associated contaminants into the lagoon. In that sense, different environmental regulation exists which try to guarantee the environmentally safe use of wastewaters, and this is very important in the area because up to 98% of the treated wastewaters generated are reused in agriculture. So this is when the field campaigns of this thesis took place. Two field campaigns initially thought within the project, and then we took advantage of a specific flash flood event that affected the Mar Menor Lagoon, which is shown here, called as Dana from 2019. This is how the lagoon looks before and after the flood, so you can imagine that the area becomes completely flooded, and any wastewater treatment plants in the area is completely collapsed in this situation. So we suspected that this could have an effect and decided to take samples a few weeks after the flood as well. And very recently, the Mar Menor Lagoon became the first ecosystem in Europe to be granted legal personhood after a long fight by locals to protect the values of this lagoon. So this is supposed to facilitate its conservation, although the lagoon has reached a very critical situation nowadays. On the other hand, the Ebro River Delta is the mouth of the second longest river on the Iberian Peninsula, the Ebro, which flows in northwest-southeast direction. And in its mouth, it forms an important wetland area in northeast Spain, very important both ecologically and economically. For example, rice production is one of the main economic activities in the area, but also it has been subjected to different threats. For example, the limited flux of sediments towards the delta due to the construction of different barriers along the river, the flooding of the coast due to this lack of sediments, the influence of diverse local sources of pollution, and biological invasions. For example, different alien species have colonized the area in recent times, and this has implications not only ecologically, but also economically, because they can affect the production of rice or fisheries. So this is an issue in the area, just to put you a bit in context. So in these two areas, a multi-residue methodology was applied based on target liquid chromatography coupled to tandem mass spectrometry to measure pharmaceuticals. This approach was applied to multiple environmental compartments from the two study areas, including water, sediments, plastic litter floating in the river, and different aquatic organisms, such as river biofilms, sea cockles, sea snails, and different species of fish. The fish were actually dissected into different tissues which were analyzed separately: fish muscle or fillet, fish plasma, and fish liver. This approach is actually quite novel because most of the existing studies focus on a single compartment, or they combine two at most. But we try to include more compartments and try to see if there's a relationship between them. So the target method, the target list, included a total of 69 compounds, and out of these, 50 pharmaceuticals were detected in our environmental samples, and they belong to 13 different therapeutic groups. The target list also included human metabolites of these pharmaceuticals, and among them, 6 were detected in the environment. One of them, for example, the metabolite of an antidepressant, which is the first metabolite that was included in the European watch list. This table tries to summarize the number of compounds that were detected in the different environmental matrices from the two study areas. Different trends can be extracted from this table, such as the fact that river water contained a widespread presence of different therapeutic groups. The presence in seawater was much more limited. Sediments turned out to be minor sinks of pharmaceuticals, although there was a tendency for compounds which were relatively hydrophobic to sorb on the sediments. And in general, aquatic organisms showed a remarkable accumulation of pharmaceuticals. In the Ever River, for example, river biofilms show the highest detection of pharmaceuticals. And in the Mar Menor, this was quite surprising because different organisms showed a relatively high presence of pharmaceuticals, and this contrasted with the levels in water, because the prevalence in seawater was quite low, and this was surprising. I will try to develop this part later on. So altogether, this indicated an added value of biomonitoring for different reasons. Firstly, because in general, it relates well with findings in other matrices, classical matrices such as water or sediments. Also because it can provide information on certain biota-specific compounds, so compounds that are generally not present in water or at low concentrations, but they can accumulate in organisms. So by grabbing only water samples, we can be missing information on these compounds that from the toxicological point of view can be relevant. And also, we found that different tissues dissected from organisms could be useful for different purposes, for example, in environmental and human risk assessment. In the comparison between liver, plasma, and muscle, while liver showed the highest levels, plasma can be interesting, for example, because it allows the non-lethal sampling of fish. You don't have to kill the fish to extract this type of sample. And muscle can be more interesting, for example, from the point of view of human health, human risk assessment, because it's the edible part of fish for consumers. Additionally, we included plastic litter as potential environmental matrix of interest. We collected floating plastics from the environment. And although the presence of pharmaceuticals adsorbed to the plastics was quite limited, we found up to 7 compounds present in these plastics, which were collected at different sites along the river, and they were characterized. Different types of polymers were represented among the samples that we took. And in some of the cases, certain pharmaceutical compounds were present at quite high levels, such as acetaminophen or paracetamol, which I'm highlighting here. So altogether, this talks about the potential of plastic litter. This is quite preliminary data, but seems to indicate that there's a potential of plastic litter to act as passive sampler of some pharmaceuticals. So now I will try to show some trends in the presence of pharmaceuticals, geographical and seasonal trends, focusing on the specific case of the Ebro River Delta. This combination of figures show the geographical variation in river sites, which are in the left figures, in coastal sites, which are in the right figures, during summer in the upper part, or during winter in the bottom part. So geographically speaking, we found a similar total concentration of pharmaceuticals along the river, and this was quite surprising because the upstream of the river is a rural area, and we didn't suspect much to be released from this area. But we found out that some of the municipalities do not even have a wastewater treatment plant. So even though the population is limited, they might be contributing to this type of pollution through the discharge of non-treated effluents. On the other hand, we observed that the concentration in rivers was higher than in the receiving coastal bay, and this was kind of expected due to a dilution effect. And additionally, we thought that there was a possible contribution of certain marine-based sources, such as aquaculture activities, because we found antiparasitics, which are used in the production of fish, and this activity is quite common in the delta. Now focusing on seasonal trends, the trends were quite weak in general, but we saw a tendency for higher levels during the winter season. And there are different factors that could explain the seasonality in the presence of pharmaceuticals, such as differences in consumption patterns, for example, for some compounds between winter and summer, differences in the efficiency of treatment technologies between summer and winter, or another factor which we think can be relevant in this study is the higher degradation, natural degradation of these compounds, which can occur during summer due to the influence of higher sun irradiances and temperatures, which can boost phototransformation reactions or biodegradation activities in the environment. So as we observed higher levels during winter for both river and coastal sites, we suspected these seasonal factors could explain our findings. Also, moving to the Mar Menor study case, I decided to focus some attention on the longer-term trends of pharmaceuticals by comparing the existing data. So we went back to the existing studies and chose a comparable set of sites and compounds to make a comparison between our findings and the findings that were obtained a decade ago, because we knew that in between there has been a change in terms of management of wastewaters. So looking at total concentrations, we found a sharp decrease in pharmaceutical concentrations, and we attributed this to these wastewater reuse practices that have been implemented in the lagoon since 2015 and probably have limited the direct and continuous release of wastewaters into the lagoon. Therefore, wastewater-associated contaminants might have decreased due to this measure as well. However, this data didn't match well with bioaccumulation data, because we observed that within organisms, the concentration of pharmaceuticals was quite high. So this was a bit surprising. So we thought that there can be still, that pharmaceutical pollution has not disappeared from the lagoon at all, but probably the ways that pharmaceuticals find to enter the lagoon have changed. For example, there could be higher influence of intermittent discharges. So there's not a continuous release, but under certain conditions, wastewater-associated contaminants can be released into the lagoon. And in that sense, we found interesting to see what can be the impact of a flash flood event in the release of these compounds. So we took advantage of this catastrophic event that affected the Mar Menor in 2019. We took samples just a few weeks after the flood, when it was possible to access the lagoon and take different water and sediment samples, because we suspected a short-term impact on pharmaceutical concentrations in the lagoon due to mainly sewage overflows during these episodes, and also probably due to the mobilization of certain soils, agricultural soils around the lagoon, which can be mobilized and transport associated contaminants. We observed that pharmaceuticals increased significantly after the flood, especially in surface seawater samples taken from sites near urban nuclei. During these events, a phenomenon known as vertical stratification occurs, which means basically that when the polluted fresh water coming from land, dragging the contaminants from different land-based sources, when this fresh water meets the salty water in the lagoon, it tends to form a layer due to density separation, and the water column is separated into two different layers. So most of the contaminants coming from land tend to stay in the upper part, and this is actually what we saw. There was an increase in the total concentration of pharmaceuticals, and mostly this increase was associated with the upper layer, while the lower layer showed lower levels. After this event, the concentration of certain pharmaceuticals increased quite dramatically, and some of these concentrations actually exceeded the proposed environmental quality standards, such as clarithromycin concentrations, which is an antibiotic. Now that I presented the results from the target analysis, I will focus on the comparison between target results and suspect screening results, because we know that suspect screening can have an added value, but we really wanted to know how it compares to target in terms of assessing the presence of pharmaceuticals and their possible risks in the environment. So here we will be focusing in this specific objective related to the field study that was conducted in the Ebro River Delta, where we decided to simultaneously analyze our samples through target and suspect analysis. So the workflow for suspect screening was based on the high-resolution mass spectrometry analysis of different environmental samples. Initially, the obtained data was treated with Compound Discoverer. The same software was used for the identification of suspect pharmaceuticals. Then the suspect pharmaceuticals that were identified from the different environmental matrices were merged into a single list. Then another software was used for chromatographic peak integration, and this integration provided some areas that were used to feed a semi-quantification procedure that can give us some tentative values for these substances. As we are not using reference standards in suspect screening, we can not be as accurate as in target analysis in the estimation of concentrations, but there are certain strategies that can allow us to estimate the concentrations. So the type of information that we get is known as semi-quantitative here. So basically these calculated peak areas were converted into predicted semi-quantitative concentrations. So once we applied this workflow, the first idea was to compare the semi-quantitative data obtained by suspect against the quantitative data obtained by target. So to do this, we focused on those compounds that were simultaneously detected by target and suspect analysis in the different matrices. The y-axis shows the ratio between semi-quantitative and quantitative data. The closer this ratio is to 1, the better the prediction of the semi-quantitative strategy. Actually, we used two different strategies based on the same principle of predicted ionization efficiency, with some slight differences, but actually if you see the length of these bars, they actually behaved quite similarly. And in general, the values were below 1 and between 1 and 10, which means that the semi-quantitative approach did not differ by more than 1 order of magnitude from the real concentrations. However, due to the fact that these values were mostly above 1, we could be slightly overestimating the real concentrations, and this can have certain implications for the assessment of the risks, for example, which we want to assess as well. So this table shows the total amount of risky compounds according to target and suspect approaches. As you can see, the target analysis already allowed us to determine certain risky compounds in the environment, such as 2 different antibiotics, ibuprofen or iopramide. But suspect screening allowed us to reveal additional risks in the environment. And at the same time, the risks revealed by targets, such as azithromycin or ciprofloxacin, were not determined by suspect. And this could be because when doing suspect screening, we have a wider chemical coverage, as I said, but in general, we are using instruments that can be less sensitive. So these antibiotics, which are present at very low concentrations, can be missed by suspect approaches. So in this table, I'm trying to summarize the pros and cons of target and suspect analysis according to our results. Target analysis is limited to a pre-selected amount of compounds, but as we are using reference standards, we can be more accurate about the concentrations that we are estimating, and also the environmental risk assessment can be more precise as well. We're using higher sensitivity instruments, so certain compounds present at trace levels in the environment can be detected, such as antibiotics. And on the other hand, suspect screening provides a wider chemical coverage. But as we're not using reference standards, there can be certain prediction errors associated with the approach. But due to the fact that they can reveal additional risks, they should be considered as well. The selection of one approach or the other, or their combination, always depends a bit on the objective of each study. But for this type of study, as much as possible, I would encourage a combined approach that uses suspect screening to initially determine which can be the risks in the environment, and then includes these compounds into target lists for a more accurate assessment of presence and associated risks. So we're moving to the last chapter, focused on the impact of pharmaceutical, personal care products, and microplastics combinations in the environment. So therefore, we are focusing on the bottom part of this figure through the experiment conducted on biofilms and the experiment conducted on marine mussels. The river biofilm experiment incorporated high-density polyethylene microplastics and the antimicrobial triclosan. Triclosan is not pharmaceutical because it's not used in humans, but it's used as a disinfectant for different applications. It's quite well known in terms of toxicity, but there are certain effects, such as its interaction with microplastics or its effect on the spread of antimicrobial resistance in natural communities, which are not very well known. The experiment lasted 72 hours, so it was an acute exposure experiment, which these 72 hours were preceded by a growth period of 5 weeks to obtain the biofilms, to put them in the tanks and perform the exposure part. Then the mussel experiment incorporated 2 different pharmaceuticals, the antidepressant citalopram and the lipid regulator bezafibrate, both at environmentally realistic concentrations. Also, they are relatively hydrophobic, so we suspected that they could have tendency to partition into biological tissues. This experiment lasted longer, up to 21 days, which were followed by a depuration period of 7 days in clean water. The type of microplastics that was used was very similar to the previous experiment, high-density polyethylene microplastics, and in both experiments the particles were smaller than 50 microns. And these are at the end of the experiment, we decided to measure a series of parameters in both biofilms and marine mussels. Some of the parameters that we measured were related to the bioaccumulation of microplastics and chemical contaminants. Then we measured some structural effects, such as changes in the bacterial community, and some functional effects as well, such as the metabolomic approach that we applied to marine mussels. So in the next slides, I will go through some of these endpoints that we measured. In regards to microplastic accumulation in the biofilms, we detected that the spiked microplastics, the microplastics that were spiked in the system, ended up in a relevant proportion on the biofilms. They were adsorbed on the surface of biofilms, actually not only adsorbed, but actively incorporated by the biofilm matrix by the end of the experiment. These images were obtained by scanning electron microscopy and show how the microplastic beads, these big balls here that you see, the spheres, are the plastic beads that we used, and the background is the biofilm surface. So you can see that they are deposited, and as you can see in the other image, the particles seem to start to be surrounded by the biofilm matrix. You can see different components such as diatoms and some bacteria that grow around the particles. Actually, we estimated that between 10 and 17% of the added microplastics ended up in the biofilm. And this is one of the first studies to empirically demonstrate that biofilms could be relevant sinks of microplastics in the environment. So once that we proved that the microplastics could interact with biofilms, we wanted to assess if these microplastics could have any influence on the bioaccumulation of certain chemical contaminants, in this case, the antimicrobial triclosan. So well, we saw that triclosan was remarkably accumulated in the river biofilms. Also, it was transformed by biofilms into one of its main metabolites, known as methyltriclosan. But as you can see, none of these processes seem to be affected by the presence of microplastics in the system. So it seems that although microplastics were accumulated in the biofilm, they didn't carry any significant amounts on the surface of this antimicrobial into the biofilms. Now we're moving to the mussel experiment. Mussels are known to very actively take up microplastics. So they ingested microplastics, and again, we wanted to see if this could have any effect on the intake of pharmaceuticals, in this case, the lipid regulator bezafibrate and the antidepressant citalopram. Both compounds were accumulated to a different extent because the antidepressant was much more heavily accumulated compared to the lipid regulator, up to 1,000 times higher bioaccumulation for the antidepressant. These compounds were also detected in hemolymph samples, which were used as a complement to whole tissues. But these processes, again, were not influenced in general by the ingested microplastics, as you can see by similar bars for the two compounds, similar length of the bars. But when looking at the early accumulation of the antidepressants, so at things that happened before 21 days, which was the end of the experiment, we realized that microplastics actually decreased the intake of the antidepressant. And we suspected that microplastics could be interfering with assimilation processes in the muscles. Also, by looking at the end of the depuration period, so after 21 days, the mussels were exposed to clean seawater to see how they could excrete these pharmaceuticals. And we realized that although the tissue concentrations of citalopram decreased quite sharply, by the end of depuration, they still contained a relevant amount of this antidepressant. And this can be quite relevant in terms of human risk assessment because bivalves which are consumed are typically exposed to this cleaning period after they are collected, for example, in production sites. So looking back at this review that I made on the existing studies on the effect of microplastics, we decided to put our results here to see where our dots are. And for the three compounds that we assessed using the two different models, our dots are laying around 1, which would indicate that microplastics do not have any significant effect on the transport of these chemicals into biofilms or muscles. So at the structural level, in the biofilm experiment, we realized that the exposure to the antimicrobial changed the community of bacteria. Certain bacterial groups seem to be favored by the presence of triclosan, while other groups seem to be sensitive to it. So you can see that due to the effect of triclosan, some bacterial families increased, while other families decreased. And this effect was generally regardless of microplastic co-exposure. So triclosan was the main driver of the effects in the community at the structural level. And this can have some implications for the functionality of biofilms. For example, the production of extracellular polymeric substance was decreased in the presence of triclosan, probably due to changes in composition, which are shown in this figure. In terms of antibiotic resistance, it was interesting for us to see if this antimicrobial could trigger the development of resistance within the community. There are several studies on pure cell lines, but not much is known about what's going on in natural communities. So we selected certain genes, and we saw that within the time of exposure, no relevant treatment effects occurred. However, we cannot rule out that longer exposure might lead to visible effects, because even though bacteria have a short generation time, within a few hours they can divide, they might need longer exposure times to show such cross-generational effects, to see an enhancement in the relative abundance of these antibiotic-resistant genes. At the functional level, we also saw some responses in the biofilm. For example, the photosynthetic efficiency increased in biofilms, probably due to a hormetic effect of triclosan, which has been described before for higher organisms like microalgae. So this basically means that at relatively low concentration, the chemical can have a positive effect on this parameter, while higher concentrations might lead to a negative effect. And this is probably related to the above mentioned, to the changes in community that I just mentioned before. Another functional endpoint, the activity of one enzyme, which is responsible for the degradation of organic matter, which is one of the key ecological functions of biofilms, was not affected. And actually this parameter doesn't seem to be very sensitive according to results in other studies. And finally, the metabolomic approach that was applied to the muscle, it was applied to digestive gland samples dissected from the muscles. One interesting thing that we saw here was that despite the different accumulation of the two pharmaceuticals, bezafibrate showed much lower accumulation than the antidepressant. The effects at the metabolomic levels of both compounds seem to be quite comparable. So a total amount of metabolites were altered by one, a similar total amount of compounds were altered by one or the other pharmaceutical. And this is relevant because some studies focus on either bioaccumulation or effects, and we try to combine these two type of endpoints. These are some of the main metabolic pathways that were altered by pharmaceutical exposure, which actually was the main driver of the effects. Microplastics showed some effects, but to a lower extent in the muscles. Up to around 60 metabolites could be identified, which belong to these pathways, basically. Some pathways were activated by the presence of the stressor, while others were deactivated. In general, the metabolism of muscles was therefore dysregulated by exposure. Some of the effects that were observed in relation to pharmaceutical exposure were quite well connected to the expected mode of action of this pharmaceutical in humans. So let's say muscles reacted in a similar way to what humans do when they take up these compounds. For example, bezafibrate, which is a lipid regulator, is known to act as an agonist of a cellular receptor, which has a regulatory role on energy metabolism. And in muscles, it interferes with the citrate cycle, which is also very connected with energy metabolism. Then the microplastic effects were non-specific, because microplastics rarely interfere with a specific target within the muscles, but instead they can provoke a general modulation of metabolism through the generation of oxidative stress responses. And also, interestingly, the fact that we included a depuration period allowed us to see if some of these effects that we saw were reversible. So basically, if the muscles were able to recover after exposure. And while we saw that some of the pathways were kind of recovered after depuration, some of the effects were long-lasting or even appeared after depuration, which I call latent effects. So if the committee authorizes me, I will proceed with reading the conclusions. Target analysis confirmed a widespread occurrence of pharmaceuticals in freshwater and to a lower extent in marine environments, especially in the Mar Menor, probably due to an improved wastewater management in the region. Biomonitoring provides an added value in the environmental assessment of pharmaceuticals, and the use of less invasive techniques, including river biofilms and biofluid sampling, is highly encouraged. Suspect screening constitutes a useful complementary approach to the target analysis of pharmaceuticals, but due to its possible limitations as a standalone methodology, a combined approach is recommended to guarantee an accurate assessment of occurrence and risks. Co-exposure to microplastics did not increase the bioaccumulation effects of PPCPs in mussels or biofilms, but the potential of biofilms as environmental sinks of microplastics and the effect of microplastics on metabolic pathways of mussels were highlighted. The antidepressant citalopram had a much higher bioaccumulation potential than the lipid regulator bezafibrate, but the magnitude of their effects on the metabolome of mussels was similar and stronger than microplastic effects. So I would like to thank to the different institutions that participated in this thesis, also to the funding agencies, and finally, thank you all for your attention.
Thank you very much. And now we are going to start the discussion session. And in the first place, the Dr. Maricel Gross, you have the floor.
Yeah, thank you. First of all, I would like to congratulate you and your supervisors for this amazing work. I think it's of outstanding scientific quality. I mean, also you did a great summary now with this presentation, with all the big amount of work that you have done, which is very difficult to do in just 45 minutes. I think you did a great job here. Also, I think you published many papers within this thesis. You have 5 papers, which is also an outstanding achievement within the short time frame of a thesis. And 2 of them are in very high quality, reputable journals within the field of environmental sciences. So this is something very important to acknowledge as well. Also, what I liked also is that you have tackled many different aspects within your thesis. You have worked with exposure experiments, you have done target analysis, suspect screening with a wide range of environmental matrices, working with biota, water, sediments. So I think you complemented your background. You acquired a comprehensive knowledge, which is also a very good achievement within a PhD thesis. So I would also like to congratulate you for that.
Thanks.
But okay, now it's time maybe for some questions, not only congratulations, no? I liked also very much how you structured the thesis. The thesis reads very well, and it's very well structured. But only one minor comment maybe is that in one of the sections you had this in the introduction. You had the strategies for the effects assessment, model organisms, environmental risk assessment. You placed these sections within the microplastics analysis only. But maybe this applies to also pharmaceutical analysis. No, maybe I guess that you structured it this way because you tackled these topics within the microplastics section, maybe.
The topics you mentioned, sorry, were?
In the introduction, you have these sections on 1.2.5, no? Strategies for effects assessment, the model organisms, the environmental risk assessment. Maybe these sections are general, no, for microplastics and pharmaceuticals, but you placed these sections just under the microplastics.
Yeah, actually they are quite general, because when talking about effects, for example, when I introduced the different approaches to assess effects, they can apply to different types of contaminants. They are not unique to microplastics or pharmaceuticals. So I think it went a bit with the flow of the thesis, in the sense that I started talking about pharmaceuticals, then I switched Then I covered by monitoring strategies. Then I switched to microplastics, talking about monitoring strategies and so on. And then I started to talk about effects. But it's true that maybe it could deserve a separate section as well, because I covered types of effects that can apply not only to microplastics, but to other contaminants as well.
Yeah, but this is a very minor comment because I found the thesis extremely well written, which is something very, very, very positive. Okay, in the introduction, you mentioned that during these depuration processes, that the mussels suffer, that they release an amount of microplastics, but you tackle this issue only for microplastics, but it's not mentioned for pharmaceuticals. I guess that for pharmaceuticals it's the same, that during these depuration processes, some concentrations are also released.
Yeah. The idea is that compared to other substances, actually, once organisms take up pharmaceuticals, their metabolism can be quite active, and they can either excrete the unmodified version of the pharmaceutical or change it into metabolites that are more easy to excrete. So during the depuration period that apply to the release of the antidepressant, for example, it was seen actually through high-resolution mass spectrometry that not only the mussels released the unmodified version, so the parent compound, but that the main metabolite of citalopram was generated in the process. So yeah, even if it's not developed in the introduction, of course pharmaceuticals can be metabolized by organisms, and this can affect their excretion during a depuration process.
Okay.
Actually, in this experiment, we took samples also in the clean water. So we exposed the mussels to clean water. They were allowed to depurate themselves for some time. And then we took samples from water samples by the end of that depuration period. And we saw that the levels of citalopram in water increased along this period. So this demonstrates that they are cleaning themselves. And also in that time we measured the metabolite that is produced by mussels. So in a similar way to the human metabolites that we find associated with wastewaters, we also see that bivalves, for example, or fish have metabolic capabilities to degrade pharmaceuticals. Yeah.
But they still have residual amounts of these contaminants. And do you know if these residual levels pose a risk for human consumption, for instance?
We didn't assess this directly. There is research on this field of human risk assessment going on that specifically tackles this issue, taking into account the concentrations that are found, which we show, but also they take into account the intake of different foods, which might be contaminated in different ways. So this is quite also the cooking style, because it's not the same to eat raw food and cook it. So we didn't address this complex issue directly, but I think I mentioned this in the presentation, that it's interesting that after depuration we saw this residual part, because this can apply to real case scenario where you have a production site of mussels, for example. You collect your food, your potential food. Normally, I think it goes like this: they take the mussels to a clean pool to leave them some time to depurate themselves from pathogens, for example, or toxins. But also, it would be interesting to see how efficient they are in cleaning themselves from chemical contaminants such as pharmaceuticals. We saw after depuration, at least for 7 days in clean water, that it wasn't enough for them to be clean, completely clean.
Okay, thank you.
Welcome.
Also, in the introduction, I liked it very much that you made a very good overview about the different analytical methods that are available for measuring microplastics. It was useful for me, at least, that I'm not so much into this topic. But in your opinion, I would like to hear for you what is the most effective method for characterizing microplastics, and if these methods are fully evolved, especially for analyzing complex samples like biota or sediments.
Well, it's the analysis of microplastics, and the analysis of microplastics is a hot topic nowadays, and the methods to analyze microplastics have been evolving very rapidly in the last years. They have changed since I started my PhD. In my thesis directly, we used quite basic methods, let's say, to monitor microplastics in the exposure tanks, for example, because we wanted to guarantee that we are sure about the availability of plastics for the mussels. So we used certain basic techniques like fluorimetry, because the microplastics were fluorescent. We chose fluorescent microplastics to be easier to track them in the process. But there are more advanced techniques now. At ICRA, they have a pyrolysis instrument coupled to gas chromatography, mass spectrometry. So this is one of the advanced techniques that allows you to measure not only microplastics, but also some associated contaminants to the additives, for example. Also, there have been advances in microscopy-related techniques, which allow you to not only visually inspect or explore the microplastics according to their shape, their size, and so on, but also to characterize, because they are coupled to infrared technologies that allow you to obtain compositional information about the microplastics. So and this is coupled with computational tools to quantify your microplastics automatically, so you can get a very good overview on how many microplastics you have, what are the sizes of microplastics in your filter, in your sample, what kind of polymers you have. And this is one of the most advanced tools. It's also very expensive to have one of these, but this is changing very fast, and the way we analyzed microplastics 5 years ago probably will not compare to the current analysis. No? Changing.
Of course. Okay, thanks. Also, along the thesis, and you showed us in the presentation that, you mentioned that biomonitoring can be a good tool to assess water quality, but also you showed in your results that when you analyze biofilms or mussels or fish, even in the different tissues, you have a different profile of chemical contaminants. Maybe, for instance, if you used other tools like passive samplers, for instance, could you avoid this heterogeneity, let's say, in the results? Or what do you think? I mean, could passive samplers be equal to or similar to what you have done with biomonitoring?
We didn't use directly, we didn't compare our results against passive samplers directly in this thesis, which could have been something interesting, because it's one of the approaches that can be used. You place your passive sampler in a river and they can act in a similar way to biofilms, for example, in the sense that they passively absorb certain compounds. It can be interesting. There are also, I think, limitations in the sense that depending on the type of passive sampler, the sorbent that you use, you can expect a bias towards certain compounds to be more absorbed. You need to study certain things, such as the equilibrium between how long it takes for a compound to saturate in that matrix. Things according to things I've read, but there are differences in the type of information provided by different matrices, yes, and that can be a limitation. There are not so many studies looking at the relationships between matrices. We saw that for the most abundant compounds, kind of comparable information can be obtained regardless of the matrix, but then for those compounds that are present at the lower levels, there can be some differences. For example, plasma can be more representative of relatively polar compounds that tend to stay in a liquid phase, while liver, as is a very irrigated organ, can accumulate a wider diversity of compounds. But this is something that would need to be worked on more, because really selecting the sample type is an issue. You need to be sure that sampling plasma alone is enough to cover the wide diversity of chemicals in the environment. It seems that it has good potential. According to our results, it covered several pharmaceuticals, especially the most abundant ones in the environments. But every sample type can have some drawbacks. Also, passive samplers alive. So from the ecotoxicological point of view, it can always be more interesting to include some organisms that give you information on their real, on how threatened they can be.
Yeah, so in the end, for the biomonitoring, it's still necessary to analyze all these different tissues, no, not because in one point you mentioned that maybe not to, for doing a non-invasive monitoring, no, we measure monitoring plasma alone would be enough. Maybe not. No,
I think at this
case.
stage it's still interesting to combine different matrices.
Yeah.
There are biomonitoring approaches that solely rely on the use of muscles, for example, historically used for assessing the presence of different compounds, more persistent compounds. But in the case of pharmaceuticals, there can be some drawbacks. There are benefits related with biomonitoring. They are more integrative in general than grab water samples. But yeah.
Okay. Great. Another interesting thing for me about the results is that in the biofilms, you identified a wide range of antibiotics, but you didn't find them in the sediments, while these compounds have high tendency also to solve, at least in sewage sludge, soils, etc. So I was wondering, why do you think this is? No, why did you find so many antibiotics in the biofilms?
Yeah, that was kind of surprising, more or less, because as passive samplers, laying in the bottom also, maybe in the beginning we suspected biofilms to act in a kind of similar way compared to sediments, but the reality is that sediments showed a lower retention of compounds in general. The partition into biological tissues can be driven by different factors, not only for example, the hydrophobicity of certain compounds. In general, sediments show a trend towards higher absorption of relatively hydrophobic compounds, but other factors such as the pKa, acid dissociation constant, can explain their absorption on certain matrices. For example, biofilm is known to be, the surface of biofilm is negatively charged, so certain compounds that are ionized negatively in water can have a certain tendency to be more readily absorbed to biofilms than to sediments. Also, biofilm can provide certain interesting information because they are active transforming compounds. So certain compounds that this is not the case of the antibiotic, of the parent compounds, but we found some metabolites that seem to be actually produced by the biofilms. They were only found in biofilms and not in surrounding water. So this could talk about the metabolic capabilities of the biofilms as well as a matrix. And the fact that antibiotics were found in biofilms is quite relevant, we think, in terms of potential antibiotic resistance development, because these communities are mainly composed of bacteria, microalgae. So the fact that we found these compounds there can be relevant in terms of resistance development within these communities and across the ecosystem.
Yeah, maybe it has also something to do with the composition of these biofilms.
Yeah.
Yeah.
Another interesting step would be to really characterize in field the biofilms to see what is the effect of changing composition of the biofilms in terms of retention of different compounds. There are some experimental studies on this, but it is known that depending on the thickness of the biofilm matrix, for example, they can retain more or less compounds. Also, depending on the composition, they can have a higher tendency to degrade the compounds, or
yeah, something to explore. No? It surprised me also that when you analyze plasma, you show like a range of concentrations, and you cite that some high maximum concentrations, like reaching even 88 nanogram per milliliter. No, this is quite relevant, I think. And I was wondering what implications does this could have to have such high levels in the plasma.
Now, in particular, these findings in plasma, we tried to relate these high maximum concentrations with parameters applied to humans, which is the human therapeutic plasma concentration, and this is something I mentioned in the paper, because these are the levels that you find in human plasma and are supposed to give an effect of that pharmaceutical in the plasma. So in the case of fish, we found 2 really polluted individuals, especially in the case of plasma, and different compounds. I think it was 2 antidepressants, citalopram, sertraline, and these compounds were present at very high concentrations. And actually, these concentrations are very close to the therapeutic doses used in humans. So although we didn't study directly these effects in the fish, we suspect that the fact that they are close to therapeutic concentrations very probably have an effect for the fish.
Yeah, absolutely.
Sometimes these effects can be related to, as I said, to the mode of action these pharmaceuticals have in humans, and these antidepressants can have neurotoxic effects in the fish, and this has been seen in experimental studies.
Develop some effects, exactly, yes. And also, you found a wide range of compounds also in the liver, also at relevant concentrations, no? But then if these compounds are found in this tissue in particular, they could be subject to metabolization, I guess, no?
Yeah.
I mean, it would be relevant to check maybe for phase 1, phase 2 metabolites. Did you consider that in your study, or?
Yeah.
Maybe, I guess there's no time to do everything.
We didn't go that far in the sense that we didn't specifically look for metabolites in the liver, but some of the compounds that are listed among the findings actually correspond to metabolites. It's always hard in the environment to distinguish between metabolites associated with humans and metabolites produced by the fish or the mussels. Probably you need to complement this with experimental data to remove these factors that can be confounding you. But among the target compounds that we detected in liver, some of them are actually metabolites. So we cannot rule out that these metabolites actually come from the metabolism within the liver, because this organ is very active metabolically speaking. So besides retaining compounds, it for sure changes their structure.
Yeah, exactly. This is something difficult to control just with field samples.
Yeah, but it will be interesting.
You need to complement it with laboratory controlled experiments, yes. Okay, you also mentioned that the fish that you analyzed in the Mar Menor study in particular, that is used also for human consumption.
Yeah.
Then the levels that you found, is it known if they pose a risk for the consumers, or?
Yeah, again, this would need like a more accurate assessment of risk for humans, but compared to other studies, these levels are within the low nanogram concentrations, which are comparable to what is normally found in coastal areas. But this again will depend a lot on the diet. For example, if you only eat that fish and you don't complement your diet with other stuff, then this could lead to a risk, especially to sensitive population like kids. But this is something that we didn't address. It would be a step further, take into account these concentrations that we found. Take also into account how frequent these compounds are, because it's not the same to find them everywhere than to find them in only a few fish, for example. In the case of Mar Menor, of the Ebro River study, for example, I highlighted different times in the article that concentrations, a widespread range of pharmaceuticals were found, but they were mostly associated with few polluted individuals, those that show the highest concentrations. So it's always good to take into account concentrations, frequency of occurrence, and also the diet to estimate the actual intake and see if there are any risks.
Yeah. Okay, yeah. Also, I mean, in your opinion, what bioindicator would be more suitable for the marine environment to assess water quality, mussels or fish? Because for fish, again, no, we come back to the heterogeneity, no, with these different tissues. Maybe sometimes it's not easy to
also the fact that they are moving can have an implication depending on what's the objective of your study. The fact that mussels are are sessile, they don't move, can be a benefit in certain areas when you want to study specifically what's going on in the delta. We didn't include mussels in our field studies. We performed an experimental study with mussels. It's true that we included certain bivalves, such as sea cockles in the Mar Menor, and actually in the Mar Menor they show the widest diversity, higher than in the fish. So I think bivalves, I'm not the one proving this for the first time, but this is known that in general they are good model species. So the fact that both in our experiment and in the Mar Menor they prove to be suitable sinks of contaminants makes them maybe the best option if I had to choose between one. But depending on the objectives, this can change.
Yeah, it's difficult. Also, I would like to ask you something concerning the analytical aspects, of the work. For the water, you used an in-house cartridge, combining different types of absorbing materials. But for the sediments, for the biota, for the cleanup, you use Oasis HLB. And could this affect maybe the fact that you detected different profiles of compounds, in the different samples? Could this have an effect, or did you check for recoveries, to avoid?
In the case of the target analysis, we checked for all the quality parameters, of course, and I think in the target analysis it cannot affect much, because even though we applied these two different cartridges in water versus other matrices, we always ensure that the target analytes that we wanted to check were properly recovered, and they were properly detected at low limits. So in the target analysis, I don't think it has a big effect for the purpose of our study, but it's true that the fact that we use different different cartridges in terms of suspect screening, potentially there could be different compounds that can be better extracted with the in-house cartridge, for example, which is intended to be more to account for a wider range of compounds than the Oasis HLB. This is one limitation. At the beginning, I think it was because we thought to apply this approach basically to water samples. So we decided to try with these in-house cartridges, which had been recommended, and at the end we decided to extend it to other matrices.
Well, but still, I mean, Oasis HLB is quite generic, so it covers also a wide range. So maybe it's minor also the effect that it could have. But also regarding the analysis of biota, in the introduction you mentioned many different methods that can be used, including catchers and so, and you used bit beating followed by SPE for the cleanup. And do you think that for biota, is it enough, this type of cleanup, to achieve like good extracts? Because later I see that you reconstitute your sample with methanol water. I mean, did you have any problems with this fact?
Problems in the sense of recovering the analytes, or
cleaning enough the fat from your samples?
I know that for more fatty tissues, other extraction methodologies can be applied. I forgot the
Ostro, maybe? No. Ostro plate, you mean?
No. Yeah, I forgot, but I think I have it in the introduction. The instrument we had at DICRA that is not being used anymore, next to the
gel permeation chromatography, how it's called, Yeah, but it's tricky also. I understand it's very tricky
but
to use.
In my case, I didn't work directly with very fatty acid samples, so I didn't think of applying any alternative, because in our group this bead beating methodology had been in place for a few years, and it was working well. We were adapting this according to sample type by changing the type of bead that we used, but I think in general it worked well, and the cleaning step that we applied was properly cleaning the sample. There were some matrix effects as it could be expected in any methodology, but in general I think it worked fine.
But you used internal standards to to correct for this. So no, I think all the protocol, well, all the procedures are very well. I mean, with a strict rigorosity. I mean, I don't put this in any doubt. Also, when in the Mar Menor study, no, when you compare the historical data with the data from your samplings, you saw a steep decrease, no, in the concentration of pharmaceuticals, and you account or hypothesize that this could be because there was a change in the wastewater management, no, that there is a more wastewater reuse. But are there any studies there that maybe according to this change in the wastewater reuse, more pharmaceuticals are introduced into soils or groundwater? I mean, do you know if there are any studies of this kind?
I'm sure they've been performing studies on this. I'm not sure, at least when I was writing the discussion, I didn't find any to discuss these aspects, but I think there's stuff going on, and actually, I think that would be very nice to kind of close the story. Our project was strictly, let's say, focused on what's happening inside the lagoon, but as we realized that what is happening around the lagoon has an effect in the lagoon, to close the story, it would be nice to have taken samples, for example, irrigation water samples or soil samples, to see if the sources of contamination could change from direct releases into the lagoon to more diffuse contamination sources in the soil. So for the future, I think nothing is published so far. Maybe I'm wrong, but as far as I know, nothing exists. But
this is another thesis.
things will come.
No, it's too much work. Now moving to the chapter 4, no, where you compared the target analysis with a high-resolution mass spectrometry. To what reasons would you attribute the low similarity between the pharmaceuticals that you identified with one approach and the other? Because I realized that the lower matches were with biota samples. So do you think that maybe applying other cleanup steps or something would change this result, or maybe because these samples are complex?
Yeah, this is, well, different factors could be contributing here. The semi-quantification approach that we applied, for example, relies on a calibration curve that is made with solvent. Well, we applied models to estimate based on molecular descriptors to estimate the ionization efficiency, and then this ionization efficiency is used to interpolate the slope of our compounds, and then we calculate areas based on this. So to harmonize these ionization efficiency values to the response of our instrument, we are using calibration curve. And this calibration curve was run in solvent. And in this case, because it was first approach, let's say, to this, we didn't decide to include matrix-matched calibration curves. So the discrepancy between target and suspect, for example, could be related in the estimation of concentrations, I mean, to the fact that these matrix effects weren't taken into account. Also, the fact that you have a more complex matrix can make it more difficult to see responses in biota in the same way as you see them in water, because in general, the instrument used for suspect screening has a lower sensitivity. But if besides a lower sensitivity, you add a more complex matrix, you might be missing some compounds that you see in water. Actually, I think in one of the figures of the papers, we relate the median to log ratio, which is how far is your median from the limit of quantification of your method. And those compounds with a lower median to log ratio were basically missed by the suspect screening, while those compounds that were more abundant, more visible by target, had a better match with suspect. So I think this.
But do you think that maybe it could be related also with the use of the software, no, the compound discovery software? Maybe if you used a more targeted suspect screening, no, using some databases, this could change maybe the result or the injection mode of the samples?
I think more than the treatment of data, which I think was quite refined, and also we manually inspected for after getting the raw data, we tried to see if there was any spectra related to the compounds that we detected with target. As we were not seeing them, manually see if we could find them. But more than the processing of data, I think this could be related with the analytical approach that we decided for suspect. We decided to choose a quite conservative approach because we used data-dependent analysis, and this means that only the most intense ions are fragmented in our approach. So if only the most intense ions are fragmented, one good thing is that this is quite the identification process is reliable in the sense that you have clean spectral data, but at the same time, the fact that only the most intense ions are being fragmented can make you miss those compounds that are present at lower levels and are not being fragmented. So in that sense, for example, if you want to go for a more complex approach, you could apply data-independent analysis, and in that sense, you fragment everything. It allows you to identify more things, but you need to invest more time, and it can be challenging because your fragments are mixed. You don't know what comes from what precursor. So, but these databases to play with data-independent acquisitions already exist. So that could be another thing to test. If applying other kind of approaches can lead to better matches between target and suspect.
Because now, looking at these results, if you had to choose target or suspect, what would you choose? Or do you think that they are still needed? I mean, the complementation of both techniques.
I think it always depends a lot on the objective of your study, because for some purposes it can be interesting to rely only on target. If you are imagine a wastewater treatment plant after the application of this wastewater directive, and you need to monitor 10 compounds only. I think it makes no sense to do suspect screening here. You do your target and you are with your reliable results. But if you are granted the task to perform a risk assessment in an area that you don't know, and you want to know as many pharmaceuticals as possible and to estimate the risks of these compounds, what I suggested is a combined approach based on your target list, because due to the standards that you have available in your lab, normally you work with more or less the same set of compounds that are in your target list. But the idea is that if you can fit this list of compounds with the information coming from a suspect approach, I go to the delta and I find out that there is this antiparasitic that is not included in my target list. So by doing this preliminary screening, you could end up including certain compounds that can be risky according to your first approach, and then do a more accurate assessment of its presence and risks. I think that would be my idea.
Your approach.
My approach.
Yes. Also, regarding the analysis of metabolomics data, I would like to ask you if it's challenging, no, to get to the results part, because I guess this is like treating all high-resolution mass spectrometry data, that this is very time-consuming. And I was wondering if maybe to successfully identify what pathways are altered, and so if it's important to know something about the mode of action of the contaminants, because otherwise I think it can be quite challenging, no? Or at least I'm not an expert on this field, this is why.
It's challenging, and it takes data processing, as in non-target screening, for example, is the part that takes the most time. At the end, we had the samples, and the extraction was quite simple because we wanted it to be as general as possible. You inject the samples in the instrument in a certain way. But if that took me 1 week, the processing of data took me 3 months, let's say. So it's very time consuming, and of course, if you have previous information on the mode of action, this can help you to look for your suspected metabolites in a target way, let's say. But what we did here was to apply non-target methods to look for the total amount of metabolites. And then we applied some data treatment to see firstly if different treatments separate in a principal component analysis, for example, according to treatment. Once that we saw that they were properly separated according to one of the stressors, mainly pharmaceuticals, then we proceeded to identify those altered metabolites. You focus, of course, on metabolites of interest, which are basically those that change with treatment. Potentially, we can have 5,000 metabolites in a muscle sample, for example. But among those 5,000, you will see that maybe 200 are altered by treatment. So I'm not going to identify everything. I'm going to focus on those metabolites that explain the difference between groups. And then once you have those priority metabolites, you need to look at spectral data to see if there's any good match between libraries and your obtained spectra. Also, this is part of a research study that I performed in Hydroscience, Montpellier. And in there, they have a quite extensive in-house database with information on, I think, hundreds of metabolites. So as they have injected these metabolites before, they know in which time you can expect them. So this allows a more reliable target identification of these compounds. So the idea was to complement this with the identification of some additional metabolites through this spectral comparison. But it's challenging.
And long.
It is.
Yes. Okay. Just from the last chapters of the exposure experiments, based on your results, no, you say, okay, you observed higher accumulation of contaminants. It doesn't matter if not related with the presence of microplastics, independently of microplastics. Thank you. Yeah. So, do you think that in one of the experiments you mentioned that you observed some absorption of these microplastics onto the wall, so there was like a kind of loss? So these results could be related maybe to the concentrations that you used of microplastics to maybe this loss, and also because in the last experiments when you used triclosan, you spiked at relatively high levels, no, like 20 microgram per liter. So maybe could this effect or your results are based on the concentration levels used of both compounds and to these factors?
So in that sense, the antimicrobial was spiked at relatively high concentrations, and this was not supposed to be the most environmentally realistic scenario, of course. Actually, concentrations are actually not that far from those reported in very polluted streams, let's say, but still they were high. In the case of pharmaceuticals, they were more environmentally relevant, let's say around 500 nanograms per liter. And the concentrations, of course, have the dose makes the poison. No, they say so. They, of course, have an effect on the effects that you see. Also, in the sense that microplastic availability in the system, you mentioned that there was a loss of microplastic, no, because in the setup that we decided, we found out that some microplastics could stick to the mesocosms, but we tried to control for this by allowing microplastics to equilibrate in the system for some time before we exposed the muscles. So after this equilibrium time, we started to monitor the presence of microplastics in the system throughout the process, and I think this was quite interesting from our study, because some studies do the spiking of the microplastics or of the contaminants in general, and then they measure the endpoints at the end. But I think it's important to ensure that these contaminants are actually available for the models. So in that sense, even though there was a decrease of microplastics in the beginning, we only exposed biofilms when we were sure that the concentrations were stable, and then we made this effort of monitoring throughout the experiment. And also the fact that they didn't absorb much of this chemical can depend on different factors, but the concentrations are important of both microplastics and pharmaceuticals. It could be that the higher concentration of microplastics, for example, which wouldn't be very environmentally realistic, but some of these studies that show an effect actually use higher doses of microplastics. So if there is more microplastic surface available, it could be that they can potentially absorb more compounds. Yeah, it's true.
Yeah. And now I'm coming to my last question. And okay, you evaluated this effect of microplastics with pharmaceuticals in water. But do you think that this could also, the absorption be affected, for instance, if microplastics are found in sediments? Do you know
So
if
you
there
mean
are
the
any
absorption
studies
of pharmaceuticals on microplastics that are present in...
Yeah, if microplastics are present in sediments, for instance, if if it could affect the absorption of pharmaceuticals on there. Is there any data? Do you know if there is any data on this?
I know that there are some studies performed on sediments because they are focused on benthic organisms, for example, lugworms that go through sediments to feed. And there are some studies comparing the ingestion or the uptake of chemicals with or without microplastics in the test system, for example. And I think, I don't remember the conclusions of these experiments very well, but in general, I think this effect has been questioned because absorption can happen, and there can be certain increase in the exposure to these contaminants through the ingestion of plastics. But the main reason for which this has been this is controversial is because other pathways can be more relevant. For example, in reality, mussels are not exposed only to microplastic particles in the environment. There are also natural particles which can be there, food particles. So pharmaceuticals do not only absorb to plastics, but they can absorb to other types of particles as well. So even though you find an increase in the uptake, how relevant is that compared to the intake of pharmaceuticals through food or through water? So there are studies on sediments, but I think the main controversy comes from there. Environmentally speaking, how relevant is what we see in our experiments, because there might be other pathways. No? Some studies also take this into account. We didn't include any natural particles artificially added in the experiments, but some studies decide to see, try to see what is the relevance in that sense. And I think most of the studies kind of conclude that the effect of microplastics compared to the role of natural particles or foods is relatively limited.
Okay, thank you very much.
You're welcome. Thanks.
I congratulate you again for all the work.
Thanks for the feedback.
Well, and now, Professor Fiona.
Thank you. I don't have many questions after all of that. I would like to say a really strong congratulations.
Thanks.
It was really great to get this in the post because I'm a very sad scientist. Really enjoyed the quality of the work, the rigor of the work, the level of study involved, and the conclusions that you came up with. So congratulations, that is huge. Also, I recognise that you were doing a PhD during the COVID pandemic, and I think you deserve a greater congratulations for achieving this level of laboratory work, your travel, your rigor again. So it's amazing. I suppose one thing I would like to ask you is, because there are so many disciplines in this work, could you summarize for me, because you've used the term "we" a lot in your answers, and remember, this is about you today. Could you summarize for me what it is that the disciplines that you have brought to this work, from the biological aspects, bioassay aspects, and the analytical aspects?
Well, by formation, I'm a biologist. So in the process, I had to learn a lot of things about analytical chemistry. And I enjoyed a lot learning new things. But of course, I tend to use the we, it's true. But I think this is clearly because I was not alone in this thesis. When you publish papers, you collaborate with people. And I actually, that was one of the best parts, I think, the fact that I had the opportunity to collaborate with experts in different fields, analytical chemistry, microbiology, ecology, treatments, technologies also. So I'm very grateful for this opportunity, not only with experts, but also with a lot of students, other PhD students, master, bachelor students that really helped a lot in the performance of these experiments. So the question was, what did I...
Yes. Come back to the question.
What was my
I suppose
input, let's say?
Not so much your input, but the different skills that you have brought to the entire thesis. So you've told us now you're a biologist, and you've done analytical chemistry, but there's a lot of other things that you've done in there.
Yeah. For example, in the field of analytical chemistry, you learn a lot on how to optimize your analytical methods. What are the quality parameters that you need to consider? What are the important things also when you go to the field and need to store your samples? All this stuff that I did a master in toxicology, but I didn't learn much on analytical chemistry, let's say. So everything I know now is thanks to IGRA and the people that helped me. So besides that, also there is content more related to microbiology. Also, thanks to colleagues at IGRA, I learned on how to extract samples for DNA analysis. Of course, not everything I did it myself. Some stuff was submitted to other institutes for sequencing, gene sequencing. Then I processed the data. What else? Then when I went to Hydrosciences Montpellier, they have a lot of expertise in performing exposure experiments and metabolomics. So there is where I learned about the application of these non-target strategies. Just before I had worked on the suspect data, so I had some background on this already, but the metabolomics approach that we applied, I did it by learning the methods that they have been developing there for some time. Also, in regards to treatments technology, for example, I collaborated with a colleague because we wanted to weather the microplastics, try to apply aging methodologies to see how, to try to simulate how they would look in the environment when they have been there for some time. So these colleagues work with treatment of wastewater using UV light, for example. So this is something that I was completely ignorant about, this topic before. So so I also learned from this.
So this really shows again, and all of you new PhD students, this puts a lot of pressure on you because there's a lot of disciplines in here, and I just wanted you to articulate that. So again, congratulations. I really liked the study that you did, which was almost serendipitous because of the flash flood. The study that you did in relation to that, and the data really kind of relates to the seawater. But could you, and this is a general question, could you comment, because we're seeing a lot more of these events now, the impact of those types of flash flood events on the freshwater environment, the freshwater sediment, and also the land around. What is potentially the impact from your analysis, what you found in terms of pharmaceuticals? What is the potential impact of those events from contaminants that arise as a result of those events?
Well, of course, after these kind of events, which are very critical to the lagoon and to the region, probably if you go to someone in the region and talk about pharmaceutical effects, they will say this is nothing, let's say, compared to what we are seeing now in the lagoon, because normally when these events occur, you see a huge eutrophication of the lagoon because fertilizers from the agricultural activity are discharged into the lagoon. So if you mix high availability of nutrients in the lagoon and high sun irradiance, you have these algal blooms. So normally after these flash flood events, you see how the lagoon becomes green, basically. They call it the green soup because it turns completely green due to the growth of this algae. Then as you have so much algae growing in this lagoon, at some point they don't have, they cannot grow more and they start to degrade. So they start to consume a lot of oxygen. At some point, the oxygen availability is so low that nothing can live in there. So you see mass mortality of fish. You find dead fish in the coast. So as you can imagine, when you have this scenario, pharmaceutical pollution can be a minor problem. But what we actually saw, we weren't sure about what findings what we could find, because when you have these events, you can have sewage overflows, for example, but you also have a huge amount of water coming into the lagoon. So it could be that these pharmaceuticals are actually diluted in so much water. So I think some of the concentrations that we see are quite huge, especially taking into account that they are also diluted with a lot of rainwater coming. But the fact that the levels of some contaminants are quite high, I think is quite concerning, because in an event that is not as dramatic as the one I was explaining now, higher abundance of these pharmaceuticals can be harmful for the communities. For example, I think I mentioned clarithromycin levels exceeding the environmental quality standard for this compound. So there can be effects related not to the average situation of the lagoon, but to these specific events that can mobilize also soils containing pesticides, for example. There are studies on the inputs of pesticides during these events as well. There is a ephemeral water course in this area which collects all the runoff typically, and there are several studies on what is the influence of this wady, is called, because it only carries water at certain times of the year, on the input of different contaminants, fertilizer, pesticides mainly, but also wastewater associated compounds.
Okay, thank you. I just want to go back, just something I overlooked was your criteria for selection of model organisms. In Ireland, we don't have the freedom to use fish, for example. You need to go through rigour in terms of ethical approval. What are the decisions that you need to make in terms of selecting organisms for this?
This was carried out in collaboration with ecology department from University of Girona, and they took care of asking for the authorization from the fisheries department. So I think you need to take different things into account, such as the number of fish that you are going to sample, and also the target species that you can collect. The fish were captured by electrofishing, basically, and we collected both native and non-native species.
Can I ask you a question? When you say we, did you?
Yeah, I went. I went. I was even in the boat. I was even in the boat.
Very good.
With some colleagues. Yeah.
Thank you. Yeah.
Welcome.
You referenced watch list monitoring in your introduction, use of watch list.
Yeah.
This is now an approach to selecting chemicals. What are your views on this approach to adding more chemicals in watch list and then potentially adding them to priority substance list? What do you think is the value of this, or do you have an opinion on this?
Well, I think the original idea of a watch list was to try to boost the monitoring of certain pharmaceuticals which were suspected to be dangerous for the water environment. And in the different revisions of the watch list, certain compounds are added, other compounds are removed because it is thought that the gathered data is already enough. But at some point, something has to be done about with the conclusions of these monitoring studies. I think one important step in the sense that pharmaceuticals start to be considered in regulation is this very recent regulation on wastewater, the wastewater directive, because kind of for the first time, pharmaceuticals start to be considered in regulation, in the sense that they have to be removed. I think a group of 10 indicator substances, more or less, need to be removed from influent wastewater down to 20% of the original amounts. So this is an example, I think, on how monitoring data gathered, even if pharmaceuticals were not priority contaminants in the past 10 years, the amount of information that has been gathered serves environmental regulation in a sense. Yeah.
Thank you. So I suppose that brings me on then to your approach in relation to biomonitoring. And it's really important to now look at the impacts, the risks and so on. But again, a general question is, what do we do with this information? So you have great information in your publications. We can see the risk of certain chemicals, but how does that bring us closer to changing how we monitor? So improving how we monitor. So you've got great information here from occurrence in water, in sediment, and also biological effects, modes of action, which is super. But what do we do with that information?
Well, I think one interesting thing that could be developed from here, for example, in terms of pharmaceutical effects. I try to contribute with this kind, in the case study of the muscles, for example, with mechanistic information about the mode of action of these pharmaceuticals in muscles. But I think it's not realistic to think that you can perform these studies with so many chemicals that are available nowadays, and new chemicals that will come in the future. So one interesting thing that could come from here is that if we are able to connect these molecular level effects with apical endpoints, which are useful for regulation, this can apply not only to these two pharmaceuticals that we are testing, but to other chemicals that can have a similar mode of action, for example. So I think this kind of approaches, combining molecular level responses with apical endpoints, should be important in predictive toxicology approaches, because you cannot invest time in testing every single chemical. But if you realize that some chemicals have a similar mode of action, the idea is that before a chemical becomes authorized, you could have some warning saying something like, wait, there are other studies on this same mode of action showing that this is not going to be good. So in the field of predictive toxicology, for example, I think this type of approach, this should fit this type of approaches, connecting low-level effects with endpoints that are relevant in regulation, for example.
Thank you. And when you refer to regulation, you choose fish as one matrix, for example, but the fish that we have access to in Ireland are different to those that you have here. So how then do you how do you balance the data? How do you understand, interpret the data?
I think it's always easier to work with species that are very widespread and are present worldwide. For example, certain mussels, even though there can be some differences in the species of mussel, they can belong to the same family, let's say, and they are expected to behave or feed in a similar way. In terms of fish, for example, we realized that between Mar Menor and Ebro, even though the species were not the same, we decided to compare different species or include different species that belong to the same family. And actually from, I forgot in English, but Muhilid family. And these different species from the two sites were concluded to be suitable indicators because they accumulated quite relevant amounts of these pharmaceuticals. So even though the species can be different, maybe we could extract some trends from the existing studies on which are the most relevant families, and maybe try to select some indicators within those families that can be compared across different regions.
Thank you. And how critical then is the age of the fish in all of this?
I think less critical than for other compounds, because pharmaceuticals can be integrated, can be taken up by fish. Of course, they can be retained. By compared to other chemicals, they have a faster metabolization. So, for example, when you look through the food chain, you don't see so much biomagnification through the food chain compared to other chemicals. So maybe it could be a factor to be considered, but I don't think it's as relevant as for other.
No, that's a really good answer. Thank you. Moving on, just to your microplastics work, and you showed a lovely scanning electron microscopy image of your spheres, and biofilm love surface area. So the surface area around the spheres, which you have added into your experiment, may contribute to biofilm growth. Have you considered how you actually have changed the growth pattern of the biofilm by creating this larger surface area?
You mean because as you have microplastics on the surface of biofilms, they might have higher surface area to grow on?
Yeah.
No, we were not really sure how it was going to be, because this is one of the first studies on microplastics and biofilms. So we weren't even sure if we could find any microplastics on the surface of biofilm. So it was something that we didn't consider in the beginning. Actually, I hadn't thought much about this.
I think your images showed beautiful diatoms and bacterial structures coming together on the surfaces.
Yeah.
So you're creating an environment for proliferation of growth.
Yeah.
So that potentially has a different response.
Yeah, and it mixes, because so far most of the existing studies focus on the effect of microbes on microplastics through biofouling processes. But we are looking at something more new, let's say, or less explored, that is how microplastics interact with microbes, which are already there in the bottom. But looking at the image, it seems that actually the microplastics are starting to be colonized.
Yeah.
And I don't know how this could be explored so specifically, but I imagine that the types of organisms or bacteria that grow on the plastics could differ from those on the background. So yeah, another topic that could be
Another topic, but not for you.
Okay.
No.
Thanks.
For the next one, exactly. You mentioned, and it came up in earlier questions, about biofilm being a passive sampler, and I liked the question in relation to passive samplers as the polymeric sampler. And in your response, I think you referred to, it's better to have maybe something living. But if we want to have good ways of monitoring a biofilm, tell me perhaps, why you chose that as your answer, because for me, having a passive sampler with a known structure and known chemistry is more reliable, and I can say it's more repeatable. Do you have any further thoughts maybe in terms of the answer that you gave in relation to the benefits of a biofilm compared with, for example, something like a passive sampling material?
Mm-hmm. Well, my answer was, I tried to highlight that the fact that we are using this sample type can provide kind of information that is kind of irreplaceable by passive samplers, even though I'm not very familiar with the use of passive samplers, and we didn't use them ourselves. But I imagine that in terms of reliability, if you know your solvent material, it can be very interesting to use. And compared to biofilms, compared to passive samplers, can lead to more uncertain information. But on the other hand, it can also be very interesting to see how efficient biofilms can be in trapping certain compounds that can be harmful for them. You can have certain compounds in passive samplers that are not well reflected in biofilms, or are actually biofilms do not trap them. For example, I don't know how these compounds would behave in passive samplers, but we detected that certain compounds that are found in water at very low concentrations are actually taken up by the biofilms. So without the information provided by biofilms, I don't know if passive samplers would also complement this in the same way for the compounds that we saw. But we found that as a complement to water, at least, they could provide interesting information.
Yeah, no, absolutely. And I think you're absolutely right, because passive sampling materials have not yet reached the need of pharmaceutical type compounds in the same way as they would for legacy, very hydrophobic compounds. So you're absolutely right. I just wanted to push you a little bit more. Just last question from me. You'd be glad to know, just a very small one. Why did you choose triclosan as your target for this? And your results weren't necessarily confirmatory. Why did you choose it?
It's true that triclosan is quite well known compared to other substances that we could choose. Maybe in this experiment specifically, we were more interested in the vectoring effect rather than exploring new types of effects related to triclosan. However, it is true that even though it is well known, triclosan, for example, is not much explored in terms of relationships with interactions with natural communities. It has been restricted in use from certain products already due to its known ecotoxicity. I think its presence in certain cosmetic formulations cannot exceed a given percentage. But most of the studies on antibiotic resistance development, for example, are based on cell cultures. This can be relevant because triclosan is also used in hospital environments, for example, which can be hotspots for development of resistance. But in terms of its interaction with natural communities, such as biofilms, not much is known. So I think it gave us the opportunity to study vectoring effects with a quite well-known chemical, because it's relatively hydrophobic. We suspected it to be adsorbed on the microplastics, and on the other hand, to explore certain effects such as antibiotic resistance, which was relatively unknown before this experiment.
Okay, thank you very much, and congratulations again.
Thank you a lot.
Thank you very much. Well, this is my turn. I will try. Many of the questions has been answered, and then I will have a lot of, not a lot of, only some minor comments and some question. Well, the first is only a minor comment. In the table 1, you put a column for invertebrates and another column for mollusks. Mollusks are invertebrates.
Of course.
Yes.
In table 1 from?
In table 1 from the, you remember, in page 11.
But if I remember well, in this table, I tried to separate mollusks from invertebrates, because in the invertebrate column I was referring to other invertebrates. Let's say.
I think, but well, and the other thing is also minor. Is about in the one of the paper published, you put the freshwater fish, and there is a lot of different fish, even seawater fish. But anyway, this is the past. You have published the data only for you for you taking account in any occasion. Well, now we move to the important things. Well, I think that your work is really good. My deepest congratulations for the work and also for the presentation, for the answer until now, and also my congratulations for your supervisors and your group and other people involved in this thesis. Well, you have divided in very clear way in your document. I think for me it was really nice. Also the general introduction and also the general discussion. I think you had a good work to try to all discuss all different aspects, and then and also the final part about the perspectives, future perspectives, and so on. Well, but there is a part from more analytical part, and there is another more related with the experiment and also even analytical new development or new applications. Well, about of the and even a big question. this is the field and lab experiment, or field campaign, field sampling and lab. How do you link both? Because this is the big question for many years. Field and lab, please.
In terms of organizing the thesis, for example, we wanted to take into consideration what we see in the field to build our experiments. For example, in the muscle experiment, we decided to choose 2 pharmaceuticals which according to field results seem to be relevant. For example, bezafibrate was found in, I think, in both bivalves and fish from the Mar Menor region, and the fact that it wasn't, I think it was also found in water, and the fact that this compound is relatively unknown in terms of ecotoxicity, combined with the fact that it was detected in one of our study sites, made us decide to include it in our lab experiment. That was one of the things.
In the opposite direction, from the lab to the field, do you have any idea how to
in the same experiment, for example, the fact that we chose concentrations that could be environmentally relevant and similar to those found in the environment, I think makes it realistic to the kind of effects that could be predicted in these areas. Actually, I think the current predicted no-effect concentrations for these compounds are in the same range of the concentrations that we decided to select. So I think that the type of effects that we saw thanks to the experiments, such as impact on different metabolic pathways, could be expected to occur also in marine organisms, for example, exposed to concentrations of these compounds. In the field, things are always much more complex because you have a lot of factors interacting, many different chemicals that can lead to different interactive effects, additive, synergistic, antagonistic. So of course, you don't only have citalopram or bezafibrate. So it's hard to confirm that this specific effect that we saw from citalopram will also happen in a study area with the same concentration of citalopram, because besides citalopram, you can have other compound with an antagonistic effect or synergistic effect. So according to concentrations we chose, we suspect that this could apply, for example, to marine mussels, which are wild, but things in the field in general are more complex.
Well, you had made a seasonal study to try to know the impact of in the different conditions. I would like also that to introduce or to put in your document the variations of the climate variation in that period, because in our situation of climate change, we are losing part of the seasonal effect, and then in order to compare what is the range of variation of the and also join with this idea, the reproduction. How do you think can affect to the monitoring? Because you are collecting organisms, and the reproduction is very important in order to accumulation or another factor. Do you think that this can be interesting, or what do you think?
Yeah, I think this can be quite relevant, especially for some compounds. Not sure if pharmaceuticals again would be the most relevant in that sense, because, for example, I know that certain tissues or produced during reproduction, the eggs can be quite fatty. So some compounds can have a higher tendency to stick in this fatty material, and if there's a spawning event when you release your eggs, this can be a way for some species to excrete pharmaceuticals. So the timing of sampling, when I was introducing things, I just put 2 symbols for summer and winter, because it's a type of seasonal variation. But if you're going to perform a comprehensive study on how levels change along the year, I think this is something that should be taken into account in wildlife in general, because certain contaminants can be.
Especially in the Marmel Lagoon and this area, because the variation are shorter than probably than here, because in the temperature, I think. Well, part of your work is related with microplastic, many at least from experimental work. You are work with polyethylene microplastic. What is the decision to only to work with these microplastics?
There are plenty of polymers available, which we could have chosen. Also, within each type of polymer, different types of different sizes, because anything below 5 millimeters is considered a microplastic. So potentially, you could work with any. We decided to kind of fix the type of microplastic and then combine it with different chemical stressors in the two experiments, try to reduce the variation due to the type of plastics also. It's true that if we are going to perform more comprehensive study of triclosan, effects on triclosan, it would have been very interesting to vary the type of microplastic, because each type of microplastic can have a different surface, therefore a different affinity for different compounds. So it was not the main aim of our study. We decided to choose polyethylene because it's simply one of the most widespread polymer types in the environment, and then vary all the other parameters in terms of chemical selection, for example, to see if they could have an effect.
For several theses.
Exactly.
Not for
exactly.
you. Well, only mention probably the last one question. Yes, this is my last question. You are working with metabolomic, and you are speaking about the other technique, omic technique, and what is your opinion of the advantage or disadvantage of metabolomic against metagenomics or metaproteomics?
The good thing of these approaches is that they can be related. Some studies actually combine them in their approach. I think a good part of metabolomics is that it reflects the final downstream products of all the different omics, because genomics about genes, proteomics about proteins. So genes and genes are transcribed, and then the transcripts are translated into proteins. No? So these proteins participate in metabolic pathways, which at the end reflect how the downstream products of metabolic reactions behave. So in that sense, I think metabolomics can be quite integrative about omic level effects. But of course, to be more sure on what kind of effects our stressors are triggered, the more omic tools you combine, the more the more reliable your data can be in terms of mode of action, for example. So metabolomics can integrate well. Any effects, for example, on the transcription of genes or on the production of proteins, because they are the final downstream products of metabolic pathways.
Okay, no more questions. Thank you very much. And now, please, I would like if you leave the room to discuss the member of this commission, and then we.
Thanks a lot. Eso es lo que he podido.
Yes. in this moment. Excellent. Thank you very much. Congratulations again for you and for your supervisor. Congratulations.